All right, microbiology class. We're here in chapter 3, which is going to cover two domains of life, the bacteria and the archaea. And I'll tell you this, we do not do the archaea justice. I don't take personal responsibility for that. We don't know a lot about archaea, and there's actually very few, as you guys probably saw in your homework, that, at least as far as we know, influence human health. So we're really going to focus on the bacteria here today, and then we'll talk a little bit about the archaea at the end. Chapter 5 covers the eukarya, and so we'll head there next. So as we're thinking about, see if I can figure out how to run the dual monitor here. Yay. Okay, so this table I thought was really nice because if you're a more linear thinker, I like to think in tables a lot. This looks at the three domains of life and then tries to point out kind of some of the key similarities and differences. So we'll talk about these a little bit as we go along, but something you might want to come back and refer to. Now, what I would say, kind of the big thing that people are always talking about when we think about bacteria is that they do not have a membrane-bound nucleus, right? Or organelles, right? So that's when people say, oh, there's no nucleus. That's what we mean, right? There's no membrane-bound nucleus. So that's a big one. I would also say one of the things that we'll point out often, I gotta do it over here. One of the things that we point out often, right, is that typically the bacterial chromosome, there's just one, where we're used to, say in humans, 32 pair of chromosomes. And the bacterial chromosome is also circular, where we are used to linear chromosomes here in humans, right, in the eukarya. And so there's a few things here that are really different. This is not going to mean much to you at the moment, but the ribosome in a bacteria is slightly different, 70S, right, as opposed to the 80S ribosome in eukaryotes. At this point, you don't know what that means. Just realize that they are slightly different. This is going to allow us actually to use some, say, antibiotics and things against the ribosome of bacteria without really much concern about harming our human hosts. Okay, what else do I need to point out to you? Those are probably the big ones. Oh, one other big thing here is peptidoglycan. We'll talk about this extensively, but this is what the cell wall of bacteria is made of, and this is unique to the bacterial domain. We don't see, as you can tell here, we don't see, I'm not pointing at that, we don't see peptidoglycan in archaea or eukarya. Thank you guys for bearing with me. Okay, moving on here. Okay, so when we're thinking about bacteria, there is a huge diversity of bacteria. Scientists are still researching, finding new bacteria all the time. We've probably just barely touched the surface of the numbers and types and diversity of bacteria out there. But when we're working with these in the lab, this is difficult. One of the things we're going to use to tell them apart is their shape, but these are microscopic. So even though we're going to magnify these guys 1,000 times under our microscopes, sometimes this can still be very difficult to distinguish. But a few things here to keep in mind. So some bacteria are going to be spherical in shape. And a few of these, this is review from 111, right? So a coccus is going to be a spherical bacteria, or cocci is plural. Then we move on, and again, this is one you probably heard in Gen Bio, the bacillus. This is a rod-shaped bacteria, but notice some of these rod shapes, like these guys right here, are like remarkably spherical looking. Anyway, there is, and probably the other one you heard in Gen Bio is spirillum. And so this is kind of a spiral-shaped bacteria. We'll see all of those. Adding a few more here as we move into microbiology, this Vibrio is they describe it as kind of a comma-shaped bacteria. Vibrio cholerae is the causative organism of cholera. We'll talk about that one a little bit. Spirochetes, the famous spirochete is the one that causes syphilis. So it looks an awful lot like a spirillum, right? But it's a little more spring-shaped. So maybe the coil is a little tighter, something like that. Branching filaments, we're not going to get into. Okay, so definitely a diversity of shapes. Another thing that you're going to use as you try to distinguish bacteria is their arrangement. So this is, if you think about how bacteria multiply, right, they are just undergoing binary fission. You're taking one bacteria and splitting it into two, and then each of those grow large enough and they're going to split into two again. Depending on if they only split along one plane or if they can actually split in multiple planes, you'll end up seeing different arrangements depending on which bacteria you're looking at. So for example, here we see a Diplococcus, right? That bacteria split in one plane and it starts to kind of form this, this little pair. And then notice Streptococcus, which is a term you're probably familiar with, like this is what causes strep throat. It forms a chain. So if you see a chain of spherical bacteria, pretty likely that that is a Streptococcus. Don't freak out, that's okay. We're in the lab, we can do this. But notice that they can divide differently as well. So you take that same shape of bacteria, and here's another really common word, right? You've probably heard of Staphylococcus, right? Or a staph infection, or MRSA, which is methicillin-resistant Staphylococcus aureus, right? When you look at this bacteria microscopically, it's actually divided in several planes, and so it ends up making this big cluster. It kind of looks like a bunch of grapes, right? So this arrangement can be really distinctive and help you identify bacteria. If you're dividing in 4 planes, you might get a tetrad or a sarcina, right, if it forms like a little cube. Bacilli, you'll get the same kind of thing. Here's a rod-shaped bacteria dividing in 2. Sorry, that's a little hard for you to read probably. Diplobacilli, streptobacilli, there's that chain. Now, if the plane it divides in, instead of them being this way, it ends up this way, it's considered a palisade arrangement. And again, this can help us distinguish different bacteria. This book, it's called Berge's Manual. This page looks insane to you right now, but basically, this is some of the stuff we'll be doing in lab, not near this crazy, take a deep breath, but this book basically lays out how you distinguish bacteria. So some it might be as simple as a shape or arrangement or how it stains, but then they start getting into some real nitty-gritty here. Certain enzymes maybe that it has, or what sorts of molecules it can break down, or if it can survive in certain pHs or salt levels, or what temperature it grows at. Okay, and so we will be running a ton of these tests. This is a lot of the stuff that we'll be doing in lab. Okay, so a lot of what this chapter goes through is kind of that standard, what does your typical bacteria look like? What structures does it have? So we are going to run through this actually fairly quickly, and we're just going to kind of use this as our outline here. So we're going to start on the very outside of the cell and work our way in, okay? And see what kind of things we come across. So if we start on the very, very outside, we're going to do what are called the appendages. So like on you, that's your arms and legs. And so what are these appendages, these things sticking out on the outside surface of a bacterium? We have flagella, pili, and fimbriae. So let's take a look at those. Okay, so flagella you're probably familiar with, right? Think about in a human cell, the sperm, right? It has the flagellum. This helps it swim. Same thing going on here in bacteria. Not all bacteria have them, but some do. And since this is microscopic, it can be really hard to see the flagellum, but you can often tell if a bacteria is moving through a media, a jello, an agar that we're trying to grow it in. That can be a really good indication that it does in fact have a flagellum. Okay, we're not going to go into the details here of the anatomical structure, but basically what you'll find is it ends up being all the spirilla have a flagellum, something like half of the bacilli, and really just a few of the coccus are going to have this appendage. So here we go. This can look different. This can help us identify bacteria as well. How many flagella there are, what the arrangement of those are. And so over here for A, that is called a monotrichous arrangement. You'll see all these terms in your text, right? So A is our monotrichous, mono being one. Trying to get my pen back here. There it is, right? So a single flagellum. This next one over is called lophotrichous, and the prefix loph just means a tuft. So there's a little tuft of them. Amphitrichous, it has one on each end, right? So an amphibian lives on land and water. It's amphi, it's both. So there's a flagellum on both ends here. And then this guy is peritrichous, meaning it's got flagella basically all over the surface, right? The whole perimeter, peritrichous, has flagella. Okay, there's also some strange arrangements. There's one where the flagellum is kind of like a barber pole around the cell, kind of between the cell membrane and the cell wall. That's called a periplasmic flagellum. Anyway, lots of different options for how this can work. But regardless of which of these arrangements, what we'll see is that these are important in movement. So bacteria are going to use chemotaxis, which is a term you should be familiar with. So think about it as movement towards or away from a chemical stimulus. So ideally, we might see bacteria moving towards, say, a nutrient. So in this case, if they're showing this is an attracting concentration, so maybe that's a nutrient, right? Or we might see bacteria kind of running away from a toxin. Either way, that would be chemotaxis. It's just a matter of whether it's positive or negative chemotaxis. Now, the way bacteria do this is kind of funny. So they don't have a whole lot of control over this movement. So basically they can turn their flagella on and move in whatever direction they're pointing, or they stop beating their flagella and then they just kind of tumble through whatever their environment is. And so what this image is showing you is a series of what we call runs and tumbles, right? So the run is, I'm going for it, right? Where's that nutrient? Okay. And then it'll stop and the bacteria tumbles. We're not sure which way we're facing and we're going to start again. So notice what happens. So in this case, right, they're showing there's an attractant involved here. So notice that the runs that are headed in the right direction actually last much longer than, say, like this run right here, which was actually moving away from the nutrients. It was really short. So we don't have a ton of control, but we can tell we're going the wrong way, so we stop. And hopefully as we kind of tumble, we'll end up facing the right way and we'll start again. So that's a little different than, say, like sperm swimming in a certain direction using it's still chemotaxis, right, towards a stimulus maybe that the egg is producing or something about the female reproductive tract. So bacteria, not quite as fancy as far as this goes. I thought that was kind of fun. And so then notice over here, right, when there's no attractant, those runs are basically the same length regardless of which direction. And that kind of makes sense. Okay. So another appendage is what are called the fimbriae. These are essentially like little hairs, almost like little Velcro, all over the surface of this bacterial cell. That's going to be serving to help this bacteria in attachment. So bacteria might want to stick to a surface say a catheter. They might want to stick to each other. We'll talk towards the end of class about something called a biofilm, and this is in fact when bacteria kind of stick to a surface, stick together, and actually start working together. And this can actually be a really important component of bacteria's ability to colonize a host. So you think about something like a urinary tract infection. In order for that bacteria to travel up the urethra, right, urine is trying to like flush it out. And so it's going to use things like fimbriae to stick to that surface. Okay, and then pili, or the singular would be pilus. So this is going to be a elongate, rigid, hollow tube that the bacteria puts out, sticks into a neighboring bacteria, and delivers DNA. Might sound familiar. This is basically this is for bacterial conjugation. So this is for, right, a conjugal visit. Now you're sitting there going, wait a second, bacteria, they only do asexual reproduction. It's true, but bacteria are a lot more promiscuous than we talk about in Gen Bio, right? So they are actually putting out tubes, delivering DNA to each other. These are particularly common in gram-negative bacteria, and we'll talk about gram-negative, gram-positive, but this is a really big thing in things like antibiotic resistance. If you have the gene for how to survive, say, penicillin, you are able to hand the genetic instructions for how to survive penicillin to your neighbor. And so we can see those resistance genes actually moving through even unrelated bacteria. So keep this one in mind. This is kind of wild. Okay, so we're going to keep moving in. Those are our three appendages, right? The fimbriae, the pili, and I'm flagellum, totally blanking there. Okay, so those are your appendages. So we'll keep moving in a little bit here and kind of look at the surface coatings of the cell. So again, if you're kind of thinking about the inside of the cell, we see in blue here the cell membrane, and then we move out to the cell wall. And then what we're going to talk about as surface coatings here are two different layers. This S layer is made out of a single type of protein, and it's linked together, and it almost ends up being kind of like a chainmail. This is absolutely for protection. Maybe it plays a role in attachment. The glycocalyx. This is going to be a coating of polysaccharides, right? So complex sugars, or glycoproteins, proteins with sugars attached to them. And this is going to be important both in protection and attachment as well. So this glycocalyx comes in two main arrangements. It can be kind of set up more like a slime, they'll call it the slime layer, or it can be as what we call a capsule. So if it's a fairly loose arrangement, this glycocalyx, all these sugars or glycoproteins, right, end up forming kind of this slime. So I don't know if you can see on the outside of these little bacterial cells, or here this is an actual microscopic image of all those sugars. Okay, this is super important in things like biofilm production. So that's what they're trying to show you up here, is this whole layer of glycocalyx. Maybe it's attracting other bacteria in, etc. If the arrangement of the glycocalyx is tighter around the bacteria, what we'll get is this capsule. And so that light area is the capsule. That's just the way it's stained. And this is really common, or more common, in pathogenic bacteria that are going to cause illness. It seems to protect the bacteria against phagocytosis. So it's preventing, say, your macrophages or your neutrophils from actually engulfing this bacteria. Okay. So then that's basically what's going on the outside of the cell, right? The appendages and the different surface layers there. So we're going to move into this region that's called the cell envelope. And basically what we're looking at here are all cells having a cell membrane and a cell wall. And then outer membrane is in parentheses because some bacteria are going to have this outer membrane. Okay, so what does this look like? So we have two different arrangements of this cell envelope, and these look different depending on if you're looking at a gram-positive bacterium or a gram-negative bacterium. But what we have going on here, so we'll start with the gram-positive bacteria. Here, and I realize this is tiny for you, hopefully you're looking along in your book. Here on the inside, and we see this on both sides, right? On either cell type, is that cell membrane, right? Phospholipid bilayer, some embedded proteins, you guys should recognize this. That's your cell membrane. Over here in the gram-positive, we then have this huge, thick peptidoglycan cell wall. Okay, so this cell wall, this peptidoglycan, and sorry, on the gram-negative, it's just this thin little layer of peptidoglycan. Okay, this makes more sense. I'll just talk about them both at the same time. And then gram-negatives, they don't have very much of a cell wall, but they do have that outer membrane. Okay, so the only bacteria that have an outer membrane as part of the cell envelope are gram-negative bacteria. Okay, so in gram-positive, you have the cell membrane and a thick peptidoglycan cell wall. In gram-negative, here's our cell membrane, a thin peptidoglycan cell wall, and then an outer membrane. So peptidoglycan, you're going to hear that word a ton. You're going to get used to saying it. I know it's a strange one. Peptidoglycan, basically they're comparing it to a chain link fence, right? But what we've got going on, basically glycan is sugar, right? You might recognize that structure as looking like a sugar from Gen Bio. And then what's connecting it in these yellow lines that they're showing, these, or what they're calling a peptide crosslink. If you zoom in and you look at this peptide, alanine, glutamate, lysine, does anyone recognize those? They're amino acids, right? And so what we end up with in peptidoglycan are sugars and proteins, and they get hooked together in this repeating fashion. So peptidoglycan is going to be super important in maintaining the shape of our bacterial cells, providing structural support, right? They don't have a skeleton or anything. So this is their structural support. And then also it plays a huge role in protecting the cell from osmotic stress. This is huge. This is probably the most important thing to think about the cell wall in a bacterium. It prevents osmotic stress, right? So if you think about there being a bunch of water, that is all the writing you're getting out of me. There's H2O. If you're thinking about there being water surrounding our bacteria here, right? If there's any kind of salt or something inside of the cell, which would be very normal, and this cell is in fresh water, if you think about osmosis, water wants to rush into the cell, right? But we said one of the problems is as water keeps coming into the cell, eventually the cell is going to want to explode. But this chain link fence surrounding the entire bacteria means that it just fills, and then the cell wall prevents that lysis. And so bacteria actually can do pretty well in hypotonic solutions, right? They're not going to rupture. Okay, so what we end up doing though is this difference between the way that cell walls are laid out is going to be a major distinguishing factor in bacteria. We tend to split bacteria into gram-positive and gram-negative. Now, we'll do some gram staining in lab so you can see this in person and figure out how this works. But I'll just say this, right? A gram-positive bacterium, at least for now, right? When we go through this process of staining it, it ends up looking purple. Purple, positive, thick peptidoglycan. Purple, positive peptidoglycan. Okay, a gram-negative bacterium, when we go through this whole process of gram staining, it ends up kind of reddish, right? And so gram-negative cells, remember they just had that, wrong way. They just had that little tiny layer of peptidoglycan. And so the purple stain doesn't stay in it as well, right? And so it's going to end up looking red. That's gram-negative. We'll talk a lot more about these distinctions, okay? But again, I pulled these. I thought this was a pretty cool picture. I'm actually showing you the cell membrane here, and then that peptidoglycan cell wall. This is an actual microscopic image here. Okay, and then same here, cell membrane, just a little bit of peptidoglycan, and then that outer membrane. Okay. Before I move on from cell walls, one other thing I do want to point out, do you see this space between the cell membrane and the cell wall? Right in there? That's called the periplasmic space. Let me go back to this picture here. So it's really hard to see, but they point out the periplasmic space here. In a gram-negative bacteria, you actually end up with two. The space between the cell membrane and the cell wall, and then the cell wall and the outer membrane. In gram-positive bacteria, you just have the one between the cell membrane and the cell wall. Okay, so this periplasmic space, this ends up being important because this is where the cell does a lot of its reactions. So think about it. This bacteria does not have a mitochondrion with an inner and outer membrane where it can pump hydrogen ions to come through ATP synthase. Remember that whole deal? It's going to do this across its cell membrane. So it's actually going to do things like pump hydrogens into this little periplasmic space, and then they can come flowing back through here, if that's an ATP synthase, into the cell. So they use the periplasmic space for reactions. Okay. Well, I was going to ask if anyone had questions, but that doesn't work here. Okay, so the cell wall, some of that's probably a little new for you. The cell membrane should sound super familiar, right? Phospholipid bilayer, embedded proteins. Anybody remember the function of the cell membrane? Selective permeability. Good. I bet a lot of you got that. Selective permeability. So these proteins really do help us decide kind of what goes in and out of the cell, and we get to make some choices there. And then, as we were also just talking, right, by using that periplasmic space, anything that we kind of bring across the cell membrane, this can be involved in a lot of different reactions. Okay, so that's the cell envelope. Again, this outer membrane is in which kind of bacteria? Gram-negative. Okay, so just a couple organelles to talk about, and then we'll wrap this up. Okay, so as we move inside of the cell, we'll run through this list, but we're going to keep it pretty brief. So the cytoplasm is the first thing on the list here. It's very similar to cytoplasm in eukaryotic cells that you probably talked about in Gen Bio. It's something like 75% water. Then you're going to find lots of sugars, amino acids, salts, right? Basically any of the building blocks or the fuels that the cell needs are going to be dissolved in this water. Okay, moving on. Ribosomes. So you hopefully will remember the ribosome as the site of protein synthesis, right? It's this little factory. It comes in two separate parts that they're calling subunits, and those are then going to link together. They read the messenger RNA and are going to produce a polypeptide chain. Again, I mentioned earlier, the ribosome here is slightly different. So we're talking about bacteria right now. Right, and so bacteria have a 70S ribosome. The S is just like a measurement, like a density measurement. Eukaryotic cells are 80S, so slightly different. But they do the same thing, protein factories. Okay, bacteria have an internal structure called an inclusion or an inclusion body. And these are often just little storage sacs. So probably the most common thing, got a little star going here, the most common thing that we're storing are nutrients. And so in this picture, they've actually, they've colored that in for you, but they stained this and they were able to see that this is essentially sugars being stored in these inclusion bodies. But then it gets kind of interesting. Sometimes these inclusions are actually some different gases. And a lot of times this is for buoyancy. So if you can hold on to a gas, like say the byproduct of photosynthesis, which is oxygen that could help you float, stay near the surface of the water where there's more sunlight for photosynthesis, right? That sort of thing. And then this is the craziest one, right? So sometimes they're storing inorganic materials, in this case, iron, and they're actually using this as kind of a magnet. So they're using this to do something telling with directionality. I'm not really sure if we know what they do with that information, right? But they can store all sorts of different things. Sometimes you will also see, I wish I could type in here. Man, okay. You will see what are called BMCs. That didn't work at all. BMC. So a BMC is a bacterial microcompartment, and these are going to be little protein chambers filled with things like enzymes, and they basically use these to run different biochemical pathways. So kind of an interesting, they don't have membrane-bound organelles, but they're using proteins to make little chambers to do some of the things that we might accomplish in some of our organelles. Okay. All right, no nucleus in bacteria, but they have what's called a nucleoid. So in this picture, this is the genetic material, and we're watching a cell here divide into two. So you're seeing new cell walls form, and then nuclear material is actually going to split. I guess I can't call it nuclear material, DNA. And so the nucleoid is this area where the DNA hangs out, but since it doesn't have a membrane, it doesn't have a nuclear envelope, we can't call it a nucleus. It's the same. It's where the DNA hangs out, right? Okay, so, and we had talked about that, again, one chromosome in bacteria, and it's usually a circular rather than a linear chromosome. You'll also find, right, so if you think about the nucleoid, right, you have this one bacterial chromosome, it's circular, hanging out in there. You'll also find outside of that nucleoid little tiny rings of DNA called plasmids, and we will talk about those more this semester. They're essentially extra chromosomal DNA that's outside of the chromosome. So tuck that one in the back of your mind. It's called a plasmid. Oh boy. You have to work on that. That's a D, plasmid. Okay. Last couple things here. Cytoskeleton. Basically, this is going to be similar to what we see in eukaryotes. It provides a little bit of cell shape. It helps stabilize the cell. Okay, bacteria can make a structure called an endospore. And endospores are dormant bodies that survive extreme environments. Like, these are the hardiest of all life forms. They can survive outer space, extreme heat, drought. You can freeze them, right? That's an endospore. It's this dormant body. This is really common in some of the genera that you might see this in are some of the bacilli and Clostridium. So if you think about, this is what causes things like botulism. This is what causes anthrax. This is what causes, and botulism is like in canned goods, if those go bad. Tetanus. So anyway, these endospores lie dormant, and then if they get into your body, they can start reproducing and can be problematic. So let's see here. This is kind of what it looks like. So basically in bacteria, we have two different stages of the life cycle. So when a bacterium is just active, doing its thing, metabolizing, being a bacteria, we would say this is its vegetative form. It's metabolically active and it's growing. But as the environment changes, right? And often as the environment changes, and particularly for the worse, this is a signal to the bacteria like, hey, you might not survive this. Why not go ahead and put your DNA, wrap it up tight in this endospore, and then we can wait for conditions to improve. And so this forms in response to environmental signals. Typically, that's a decrease in nutrients, especially amino acids. But okay, so this is what's going on here. So they're pointing out the chromosome duplicates and gets separated. And so here we see the copy of the DNA that's essentially going to get wrapped up nice and tight here, right? Covered in layers and layers, right? And wow, layers and layers until it is an endospore. So the rest of the cell is going to die from the lack of amino acids there, and we have a spore. Now that spore can eventually germinate, right? So they're showing the same size arrow here, but this could be years, decades, right? Like whatever, grandma's basement and some canned goods. That spore is going to germinate, right? It hatches out and it can go back to being a vegetative cell, a normal bacterial cell. Okay. Spores are the reason, and you'll see this in lab as well, spores are the reason we use an autoclave, or when you're canning, why you use a pressure cooker. An autoclave is just a fancy pressure cooker, but basically all of your lab materials, Patty or I puts it in the autoclave at 121 degrees Celsius, so that's over the boiling point of water, and we're going to do that for 15-20 minutes to make sure that those boiling temperatures would kill all the vegetative forms of bacteria, but you actually need steam under pressure to actually kill these endospores. That is one thing that kills them, fortunately. Okay, so to wrap up our thoughts on bacteria, I do want to mention biofilms. Now, this is also going to be the topic of your first paper, and so you can look in Canvas for details on that. You're not going to repeat what I'm about to tell you. I just want to give you a little bit of an introduction in what you're thinking about here. So basically what happens in a biofilm is we have free floating is not even the term, free living bacteria in the environment, right? Some of them start to actually stick to a surface, and then other bacteria can stick to their glycocalyx. And so we start kind of forming layers and layers of microbes. And the glycocalyx kind of all is together and forms what we call a matrix. So it's just kind of this jelly soup, and they're able to share through that matrix. Now, what makes bacteria really that form biofilms particularly interesting is a lot of people think of this as like an ancient adaptive survival strategy by microbes, so that all of the bacteria in this film are getting those getting their needs met, right? In a multicellular organism, a skin cell doesn't have to worry about oxygen exchange, right? It's getting that through the lungs, and the blood carries the oxygen, all that kind of thing. But bacteria, as single-celled organisms, they don't have a great way to do homeostasis, and it looks like biofilms is kind of a creative way to get there. This is the craziest part about biofilms. They're talking to each other. So what ends up happening here, I should use the pointer. So this is trying to show a biofilm formation. So you have your free-floating bacteria. It comes in, it sticks to the surface. Everybody's sticking to each other. Here's that matrix. Each of the bacteria starts releasing what's called an inducer molecule. So that's these little purple spheres here. This is essentially chemical communication between cells. This lets them know when they've reached critical mass. So the key word here is quorum. This is a term from government. You have to have enough members of a committee to vote. That's quorum. But in bacteria, they're basically kind of taking roll call here with these inducer molecules. And when they realize they have enough cells, these inducer molecules actually cause a change, basically change the gene expression. They cause the production of this protein, which very often is something like an enzyme. And so in mass, all these bacteria in the biofilm suddenly produce this enzyme, and they can do together what they never could have done on their own. So classic example of a biofilm is like the plaque on your teeth, right? The reason the dentist is in there scraping, or the hygienist is in there scraping it off, is because if that biofilm hits quorum, and they all produce an enzyme at once, they can actually drill through enamel, which is one of the hardest known substances, right? A single bacterium could never pull that off, but a biofilm is this adaptive strategy that lets them do this. So biofilms, I think, are pretty fascinating, so you get to write about them. Check out the assignment and start working on that paper. Okay, brief moment for Archea. As promised, brief. So Archaea, this whole other domain of life, for a long time, bacteria and Archaea were kind of squished together. We didn't have a good understanding of how they differed. Now we're really figuring out, hey, they have different membrane lipids. The cell wall is made of different things. There's different genetic sequences, etc. But regardless, these Archaea are typically considered extremophiles, meaning they love extreme environments, right? Things like thermal hot springs in Yellowstone, right? They survive temperatures no other life could handle. There's a group called the halophiles that love salty environments. So here we have the Bonneville salt flats. There's a group called methanogens that actually do some strange metabolic processes where they take CO2 and hydrogen and they actually make methane as part of their normal cellular metabolism. Right? That's all I'm going to say about the Archaea for now. They're the extremophiles. Okay, that is it. Thanks for bearing with me there through chapter 3. Let me know if you have questions about anything that you've read or want to talk about anything more. All right, see you guys.